Janak Dev, BE Biomedical Engineering, Sri Ramakrishna Engineering College. Sanya Yadav, BTech Biotechnology, Delhi Technological University. Shashwat Mishra, BTech Computer Science and Engineering, Trident Academy of Technology. Then myself, Visalakshi, BSc Botany Plant Biotechnology, the Standard Fireworks Rajaratnam College. Next one, please. Let's have a quick look of contents. Introduction, structure, pathogenesis, and what makes coronavirus virulent, screening and diagnosis, and then conclusion. Let me tell a crisp introduction about coronavirus. Next one, please. Next one. Coronavirus are a group of RNA-related virus. It affects both mammals and birds. In cows and pigs, it causes diarrhea after infection. In humans, it causes severe respiratory infections, and in mice, they cause hepatitis and encephalomyelitis. Next one, please. Then, in history, we are going to see the first case of birds and humans. Next one. In late 1920, the first case in chickens was found in North America. In 1960, the first case in humans was found as a mild cold, but after that it was confirmed as a COVID-19. In 2003, it was regarded as the black year for microbiologists because the spreading of coronavirus to many countries like US, Hong Kong, Vietnam, and etc. Mortality rate is nearly more than 1,000 patients. Total 8,096 patients was confirmed with coronavirus. The strain was SARS-CoV, identified in the same year. The primary host is bat, and the symptoms are severe acute infections. Next, please. In 2004 Hong Kong, out of 50 patients, 30 were confirmed with coronavirus. The strain is HCO-HKU1, identified in the year 2005. The primary host is rodents and the intermediate hosts are unknown till, and the symptoms are very mild. In 2012, it was affected Saudi Arabia. The strain is MERS-CoV, identified in the same year. Birds are the primary host and camels are the intermediate host. The symptoms are severe acute infections. Next, the discovery of coronavirus. Next one, please. The primary hosts are bats and rodents. The intermediate hosts are palmicids, camel, pangolin, and in some cases they remain unknown. Finally, they get infected to humans. These are some of the coronavirus which infects humans starting from SARS-CoV to HCO-HKU1. Then coming to etymology, next one please. This In etymology, on 12 Jan 2020, World Health Organization temporarily termed it as 2019 nCoV, and on 12 February 2020, they officially named it as COVID-19. Later on, ICTC designated it as SARS-CoV-2 based on the phylogeny and taxonomy. Next one, please. The source from which they get originated. We know that the primary host is bat and the intermediate host is pangolin, and then it finally infects human. How did they identify this source means? Next one please. In Wuhan's wet market, they sell bats and Chinese people also consume bats as food. So through that it get transmitted, they thought. But at the time of outbreak, there is no selling of bats in Wuhan's market. Then how they get spread? It means through the intermediate host pangolin, also known as scaly anteater. After that, it gets spread both in and out of China, those who have no direct contact with birds and pangolins. After that, they came into conclusion that this SARS-CoV-2 gets transmitted by human to human transmission, and finally it broke out into a pandemic. That's all about introduction. Let me hand over the session to my friends to continue further.
Hi, this is Sashwat, and we'll be talking about the structure of the virus. Uh, the virus primarily has four structural proteins or glycoproteins: the S protein, the M protein, the E, and the N protein, and also has several accessory proteins along with it. The virus itself is a single-stranded positive sense RNA, which is very similar to the 2003 SARS-CoV virus, like Visharachi has mentioned, but it is a bit larger in size, ranging from 80 to 90 nanometer. And also, this is the largest among all the RNA viruses with 30,000 base pairs. There is a lipid bilayer, which along with the nucleocapsid protects the virus itself. And the genome of the virus is encapsulated between all these proteins. This is an electron microscope based evidence of the virus itself. You can clearly see the spikes pointing out from the walls of the virus. Next, let's try to understand how the virus infects human cells in general. Like I said, this is very similar to the SARS-CoV virus and uses the same medium. It spreads through droplets of cough and sneezes. Also, the receptor is the very same ACE2 receptor. However, SARS-CoV-2 is way more efficient in identifying these receptors in the first place when compared to SARS-CoV from 2003. The S protein, which is the spike pointing out from the virus walls, is responsible for attachment with the ACE2 receptor. And then the E protein is, it is used for membrane permeability, whereas M is the most abundantly found and is important to keep the virus together, basically the virus assembly. The N protein, on the other hand, it directly binds with the N-terminal. I mean, its N-terminal is directly bound to the virus's RNA, through which it can replicate. Also, the N protein itself is phosphorylated, which causes the rapid structural changes in the protein, which increases the affinity of the virus. And this is how the virus replicates once it gets inside. Like I said, this is very good at identifying the ACE2 receptor. The spike protein that we were talking about has two protein regions, S1 and S2. And when it docks at the ACE2 receptor, the body, as an as an immune response to this, the TMP-RSS2 protease present in the host cell proteolyzes this protein into S1 and S2. The virus, on the other hand, takes advantage of this, and then the S1 region binds with the host cell receptor. Next, the gRNA present inside the virus acts as an mRNA and gets into the cell and then forms polyproteins PP1A, PP1B, and RNA-dependent RNA polymerase, helicase, and some other non-structural proteins. Then these NSP, these non-structural proteins we were talking about, they act as a replication translation complex, which form a double membrane vesicle. All these polyproteins that were formed, including RDRP, NSP, they get inside this double membrane vesicle, and they form a subgenomic RNA. The rest of the proteins that were formed earlier, S, M, and E proteins, they get into the endoplasmic reticulum Golgi intermediate compartment, and then further reshaped. At the end, the nucleocapsid that was formed earlier, that encapsulate all these other proteins, and then it forms a new virion. That is how a new virus is then formed. And in a process called exocytosis, it is again ejected out of the cell, which starts infecting other cells once out. This is a pictorial representation of the entire flow. You can see the virus entry through the ACE2 receptor, and then further polyproteins are formed. They enter the DMB, and they through the ergic they are again exited out of the cell. This is the entire story of pathogenesis, and I'll, my friend from here will continue.
So what makes coronavirus? What makes it virulent? Next slide, please. So we know that the ACE2 receptor plays a major role. Now, to understand how it infects our body, we should also know about ACE2 receptor, because it's the first site where it's going to bind and start infecting. So is ACE2 only in our nasal passage, airway passage? Are they not present in any other part of our body? If so, if they are present in other part of their body, do coronavirus affect those part also? So actually, yes, ACE2 receptors are present in lungs, arteries, heart, kidney, and intestine. So coronavirus also affects these organs, but not to a very large. It does not create a large impact on it. But understanding angiotensin converting enzyme 2, what it does means, angio means vessel. Tensin is again an enzyme that binds actin. And if you are from medical background, if you know actin, that is the thing that pulls your muscle while relaxing or while contracting. So when you relax your muscle, and again actin fibers will relax. So angiotensin converting enzyme, what it does is hydrolyzes vasoconstrictor, especially this tensin enzyme, and it helps in relaxing. So if you have a high blood pressure, you're going to have a large number of vasoconstrictors. ACE2 helps by hydrolyzing those vasoconstrictors and lowers your blood pressure. So if your ACE2 receptors in the cell are going to be ruptured or they're going to be damaged by the coronavirus, what is going to happen is, and if you're a high BP patient, your blood pressure is not going to be regulated because they're getting damaged, and that's why you have high mortality rate in people with high BP. Also, as I said, ACE2 receptors are not only present in the lung airway passage, it is also present in your kidney and even in brain. Now in brain also, in cerebral cortex, hypothalamus, brainstem, it is present. Does virus affect those parts of the brain also? Yes, it does. So one of the symptoms of coronavirus is that you have a loss of taste and smell, right? So olfactory part of your brain also gets affected by coronavirus, but it's not very dangerous that it will start eating up your brain. But it's still loss of senses there. Also, when you have a high viral load in your body, there are chances of this virus diffusing into bloodstream, going into other organs as kidney and heart or intestine, and you can have multiple organ failures if it's too high. Yeah. So this was explained from a paper here, as I mentioned. They studied the neurological manifestation of coronavirus. Next slide, please. So here is a picture of how badly your coronavirus can rupture the cell line. The first picture that you see is from a transmission electron microscope. This picture, the first picture, the picture A, shows like well intact cilial surface and very less disruption. This study was done to understand how virus ruptures all. So they took coronavirus samples and put it into people's nose, and they were looking at the slides, histopathological slides, and this is what they found. And in 3 days itself, if you see picture B, how badly the cell lines are disrupted. They have, uh, in just 3 days, if it could do so much damage, if it goes into lower lung also, it can cause a lot of damage. So that's why it in lungs it is highly affected, because the entry point is also very close for the virus. It does not need to diffuse all. Lung gives a good environment for it to replicate and keep growing. Yeah, so this is about how coronavirus affects our body. Next slide. I think over to Sanya now.
Thank you. So, good afternoon, everyone. I'm Sanya Yadav, second year biotech student at Delhi Technological University, and I'll be discussing about the screening and diagnosis for coronavirus. So, now the question arises, who should be tested? The current situation exemplifies the challenge of how to best utilize testing during outbreaks of novel pathogens. The initial testing criteria were too narrow to monitor and control the spread of the disease, but the sudden pivot to a far broader testing approach, even as capacity remains limited, may be an overcorrection. So once an individual has been infected by the virus, the mean time it takes to develop symptoms, that is the incubation period, is 5 to 6 days with the range of between 1 to 14 days following the exposure. Some common symptoms include fever, cough, fatigue, and shortness of breath. Some people may have other symptoms too, such as a sore throat, diarrhea, muscle aches and pains, repeated shaking with chills, or loss of smell or taste. And in some cases, individuals infected with SARS-CoV-2 may never develop symptoms, that is, asymptomatic cases. They may have very mild disease that called post-symptomatic, or they may develop severe to moderate COVID-19 disease. Next slide, please. Diagnostic testing can involve detecting the virus itself, viral RNA or antigen, or detecting the immune response to infection, that is via antibodies or any other biomarkers. Standard confirmation of acute SARS-CoV-2 infection is based on the detection of unique viral sequences by nucleic acid amplification test (NATS). The virus may be detectable in upper respiratory tract (URT) 1 to 3 days before the onset of symptoms, the presence of viral RNA in the lower respiratory tract, and for a subset of individuals in the feces increases during the second week of illness. A variety of RNA gene targets are used by different manufacturers, with most tests targeting one or more of the envelope nucleocapsid spike, RNA-dependent RNA polymerase, and ORF1 genes. Next slide, please. Serological tests have generated substantial interest as an alternative or a complement to RT-PCR in the diagnosis of acute infection, as some might be cheaper and easier to implement at the point of the care. A clear advantage of these tests over RT-PCR is that they can identify individuals previously infected by SARS-CoV-2, even if they never underwent testing while they were acutely ill. Seroconversion, that is, development of measurable antibody response after infection, has been observed to be more robust and faster in patients with severe diseases compared to those with a milder disease or asymptomatic infections. Antibodies have been detected as early as in the end of first week of illness in the fraction of patients, but can also take weeks to develop in patients with subclinical mild infection. Specimen source for antibody testing can be are mostly blood, serum, or plasma, but may include saliva, sputum, or other biological fluids. A reliable diagnosis of COVID-19 infection based on patient's antibody response will often only be possible in the recovery phase when opportunities for clinical intervention or interruption of disease transmission have passed. Next slide, please. Rapid diagnostic tests that detect the presence of SARS-CoV-2 viral protein antigens in respiratory tract specimens are being developed and commercialized. Most of these tests are lateral flow immunoassay (LFI), which are typically completed within 30 minutes. In contrast to NATs, there is no amplification of target that is detected, making antigen tests less sensitive. Commercial and non-commercial tests measuring binding antibodies such as IgG, IgM, or IgA in different combinations utilizing various techniques including LFI, enzyme-linked immunosorbent assay, and CLIA have become available. Next slide, please. Now, one or more negative test results do not necessarily rule out that the SARS-CoV-2 infection is not there. A number of factors could lead to a negative result in an infected individual, which could possibly be the poor quality of specimen because it contains too little patient material, or the specimen was collected late in the course of the disease, or it could be the specimen was taken from a body compartment that did not contain the virus at that given time. The specimen was not handled or shipped properly, or there could be any technical reasons like PCR inhibition or viral mutation. So what becomes very crucial is to test more number of people out there, isolate them and treat them. Next slide, please. So as we have come to the end of our slide, I'd like to conclude our presentation. In the past 10 years, many new coronaviruses have been identified. They infect a wide range of hosts from mammal to birds. Closely related coronaviruses have been identified in distantly related animals, suggesting recent interspecies jumps. Although the previous coronavirus, SARS-CoV and MERS-CoV epidemics raised awareness of the need of clinically available therapeutic or preventive interventions, to date, no treatment with proven efficacy are available. So far, the infected virus patient population is growing very rapidly. Effective antiviral COVID-19 candidates are essential for reducing disease severity, viral load, and transmission, thus helping to prevent the coronavirus outbreaks. Besides the development of most efficient drugs, A strategy to rapidly diagnose SARS-CoV-2 in suspected patients is also required. Thank you. That was our time. And these are the references we used in our slides. We're open for question and any other queries.
Any question, guys? I think those who have presented for COVID-19, they have variation in their presentation. So you guys should have some questions, or those participants who have not even participated in this assignment, those must have the questions or the doubts. Or everybody is sleeping.
No, ma'am.
No, ma'am.
No, ma'am.
Okay, so everything you understood by all the presentation? So, I would say it was a nice presentation again, and you have tried to incorporate all the comments I have made for earlier presentation. So it was good.
Thank you, ma'am.
And still, once check your presentation for my common comments for all the presentation, incorporate into the PPT and then resend to me.
Yes, I'm sure we will.
All right. Thank you.
That was nice presentation.
Thank you, ma'am.
Thank you, ma'am.
So, who else are ready? Narmada? Hello? Can you write if you can't speak, Narmada? So, when you guys want to present your assignment? Tomorrow, okay. So, for tomorrow, please ask me in the evening for tomorrow, because I'm not sure for tomorrow's sessions if we can arrange your presentation or not. But okay, I'll see. No problem. So now I think nobody is left apart from this group, right? Hello? Is there anyone?
Yes, ma'am.
Okay. So, I mean, I am asking you who else is want to present this assignment.